Imagine taking medicine that could make you sicker instead of better. That's a risk if drugs aren't made in clean environments. Pharmaceutical manufacturing demands the highest levels of cleanliness to prevent contamination. This is where sterile and aseptic come in. These terms are crucial for understanding how safe, effective medicines are produced. In this essay, we'll explore the difference between sterile and aseptic environments. We'll learn why they're so important in making sure drugs are safe for patients. And we'll look at common ways to keep these areas clean. Understanding these concepts is key for anyone interested in the pharmaceutical industry. It's about protecting patients and ensuring the quality of medicines. Let's start with sterile. In simple terms, sterile means completely free from all living things. This includes bacteria, viruses, fungi, and even spores, which are like tiny dormant seeds of microbes. Think of a sterile object like a blank slate with no living organism able to grow on it. Achieving sterility is a big task. It usually involves powerful methods like heat in an autoclave, which is like a superheated pressure cooker, or radiation, which uses energy waves to kill microbes. Maintaining sterility means keeping that blank slate clean and free from any contamination. This concept of sterility is central to many medical procedures. Surgical instruments, for example, must be sterile to prevent infections. In pharmaceutical manufacturing, certain products, especially injectables, need to be sterile to be safe for use. Now let's move on to aseptic. Unlike sterile, which is an absolute state, aseptic refers to a process of controlling contamination. It's about minimizing the number of microbes in an environment to make it safe for pharmaceutical production. Think of it like this. You can't make an entire factory perfectly sterile, but you can create aseptic areas within it. These areas have controlled airflow, special cleaning procedures, and trained personnel to keep contamination at an absolute minimum. Asepsis relies on a combination of techniques and practices. These include using sterilized equipment, wearing protective clothing, and following strict cleaning protocols. It's about creating a controlled environment where the risk of contamination is as low as possible. The key difference between sterile and aseptic lies in their scope. Sterility is absolute, the complete absence of life. Asepsis, on the other hand, is about control minimizing contamination to an acceptable level. You can have a sterile object within an aseptic environment, but you can't have an aseptic object within a sterile environment. Imagine a surgeon operating on a patient. The surgical instruments need to be sterile, meaning entirely free from microbes. The operating room itself doesn't need to be completely sterile, but it does need to be aseptic. This means controlling the air quality, cleaning and disinfecting surfaces, and ensuring the surgical team follows strict protocols to minimize the risk of contamination. Understanding this distinction is vital in pharma. While some products require complete sterility, many others are manufactured in a controlled aseptic environment to ensure their safety and efficacy. Section 5. Why sterility and asepsis matter in pharma. The pharmaceutical industry deals with products that enter the human body, often through sensitive routes like injections or infusions. Any contamination in these products could have serious consequences for patients, leading to infections, adverse reactions, or even death. This is why maintaining sterility and asepsis is paramount in drug manufacturing. It's about protecting patients and ensuring that medicines are safe and effective. Every step of the production process, from the raw materials to the final packaging, must adhere to strict cleanliness standards. Failure to maintain these standards can have serious repercussions. Contaminated medicines can lead to product recalls, harming patients and damaging the reputation of pharmaceutical companies. Regulatory agencies like the FDA in the United States and the EMA in Europe have strict guidelines and inspections to ensure that companies meet these critical cleanliness standards. Section 6. Real-world examples, sterile and aseptic areas. Let's look at some real-world examples to illustrate sterile and aseptic areas in the pharmaceutical industry. A sterile area could be a small, enclosed space within a laboratory where scientists handle cell cultures for drug development. These cultures are extremely sensitive to contamination, so the environment must be completely sterile to prevent any unwanted microbes from interfering with the experiments. An aseptic area, on the other hand, might be a large, controlled environment within a pharmaceutical plant where injectable drugs are filled into vials or syringes. While these areas might not be completely sterile, they have rigorous controls in place to minimize microbial contamination. These examples highlight the different levels of cleanliness required for different pharmaceutical activities. Understanding these differences is essential for designing appropriate facilities, implementing effective procedures, and ensuring the quality and safety of pharmaceutical products. Section 7. Maintaining sterility and asepsis, common practices. Maintaining sterility and asepsis in pharmaceutical environments involves a multifaceted approach. It starts with the design of the facilities themselves. Clean rooms, for example, are specifically designed to control airflow, pressure, and temperature to minimize the risk of contamination. Then comes the rigorous cleaning and disinfection protocols. These involve using specialized cleaning agents and methods to eliminate microbes from surfaces and equipment. Regular monitoring and testing are also crucial to ensure that these procedures are effective. Personal hygiene plays a critical role as well. Personnel working in these environments must undergo extensive training on gowning procedures, hand hygiene, and aseptic techniques. They wear specialized clothing, including gowns, masks, gloves, and hair covers, to prevent contamination from their bodies or clothing. Section 8. The human element, training and discipline. While technology and infrastructure are crucial for maintaining sterility and asepsis, the human element is equally important. Even the most sophisticated cleanroom can be compromised by a single breach in protocol. This is why training and discipline are paramount. Personnel working in these environments need to be constantly vigilant. They must understand the importance of their role in maintaining cleanliness and be meticulous in following procedures. Regular training and refresher courses are essential to reinforce these principles and keep everyone on the same page. It's not just about following the rules, it's about fostering a culture of quality and responsibility. Everyone involved in pharmaceutical manufacturing, from the scientists to the cleaning staff, plays a vital role in ensuring patient safety. Section 9. Conclusion: ensuring patient safety through cleanliness. In the world of pharmaceuticals, cleanliness is not just next to godliness, it's essential for patient safety. Understanding the difference between sterile and aseptic environments and the practices required to maintain them is crucial for producing safe and effective medicines. From the rigorous cleaning protocols to the specialized clothing and the constant vigilance of trained personnel, every aspect of pharmaceutical manufacturing revolves around preventing contamination. This dedication to cleanliness is a testament to the industry's commitment to patient well-being. By maintaining these high standards, the pharmaceutical industry strives to ensure that every dose of medicine is a step towards better health, not a potential source of harm.